Could you expand a bit on the mitochondrial damage that you mentioned? You talk about free radicals and you talk about mitochondrial damage. Which kind of damage? Do you know, have you characterized that?
Yeah, no, what we have done so far is just we count the number of mitochondria that has change in morphology. And when you get these cells that are infected with this Zika virus, we have basically 100% of them being with a different morphology in terms of like the formation of the crista and the organization of the mitochondria. This is the only data that I have with the mitochondria. It's an electron microscope change in morphology.
Right. Luis.
Can you tell us a little bit more about how prevalent is this incidence of microcephalia in babies born to Zika-infected mothers? Because I read a report from the health secretary in Mexico, and they say that there are over 150 boys being born from Zika-infected mothers, and none of them had microcephalia. Are there any genetic determinants that make some population more susceptible to this problem?
It's a very interesting point. Like, what can I tell you about the numbers in Brazil is that, like, when you have like around 11,000 pregnant women, between 3,000 and 6,000 of them, they got the Zika virus. I personally believe, and I have like one part of the lab working on that there are other factors involving in the prevalence of the Zika malformations in Brazil. So definitely the Zika virus is able to cause malformations, but I believe that's dependent on other factors that are more present in Brazil. I don't have more details about that, but to have a very important clue on that area.
But Stephen, so of the 6,000 infected pregnant women, you said, right?
How
many of those really develop microcephalia in the kids?
11,000 pregnant women, like we have 11,000 babies, and the number of cases confirmed 3,000, but we have up to 6,000 malformations, but like 2,000 to 3,000, they are not completely confirmed. The women, they have like the rash and all of the other characteristics of the Zika virus, but it was not clear that it was completely connected. So we'd say that like 3,000 from 11,000. Okay.
Big number. Belen, thank you.
Now it works. Following on Luis' question, is the genome of the Zika virus the same, and all infections are the same, or there could be mutations in Zika virus that produce microcephaly and others that don't produce microcephaly?
Yeah, the kind of virus that is spread in Brazil is the same that came from Asia. So there was a mutation between the original Zika virus in Africa, and then when it reached Asia, it changed a little bit. So but the virus that is in Brazil is similar, it's actually identical to the one that was in Asia. So it's much more, we believe that like could be some other factor that would make the pregnant baby more prone to have the consequence of the infection.
Conrado and then Tito.
Stevens, you mentioned 2 drugs. Have they been tested in clinical trials? Are they even combined? What is the effectiveness?
Sorry, maybe I missed your question. If the drugs that I mentioned, they are already in clinical trials, that's what you said?
If they have been tested in clinical trials to test efficacy of those drugs, and if they are given combined or just one at a time.
Yeah, they are not tested because it's very difficult to do some clinical trial with pregnant women, right? We know that like they are approved, they get used by other reasons, but the idea is that like if you have an outbreak, the idea is that this is going to be used. It's going to be very difficult to propose a clinical trial with pregnant women with their drugs. Of course, they are approved, but they are not the best thing to be done with pregnant women. But the idea is that since they are already approved by other means, and that like they seems to work not only in our model, but also in animal models, they could be one proposal to be used in these pregnant women.
Tito.
Actually, the question was the same than Conrado, but adding up, I mean, the virus is a bit different, no? The family of HCV and Zika are the same?
No, but in the case of sofosbuvir, it's going to be affecting the RNA polymerase. So this
Okay, and it does work in the same mechanism?
is working the same mechanism.
So it would work in dengue as well, or something like that?
Say it again?
Would it work in dengue, then one can imply, because Zika?
Yeah, it's interesting. It's nobody really was using it because of course the dengue has a problem, but the consequence of the dengue is different from the consequence of the Zika. So, and of course there are people working also with vaccines for both dengue and Zika. So people are not considering to use, for example, sofosbuvir in the case of dengue, but I think that the main goal in the case of dengue is the vaccine.
Juan Carlos.
Yes, very interesting. In general, viruses in the brain are recognized by toll-like receptor 3, which are located in microglia, not in astrocytes. So the neurospheres are contaminated always with microglia. Have you done any study in microglia in pure cultures?
That's a very good question. We started to differentiate microglia from iPS cells. We have very preliminary data, but in the case of the astrocytes, they have these phosphodiesterone receptors like TIM, and also the AXL. So it looks like the AXL is the main target for the Zika virus, at least in astrocytes. Okay. But definitely there is some effect of the Zika virus in microglia. We haven't done it yet. I mean, we're just differentiating the cells, but there are other groups doing this with mouse models.
Okay. Thank you very much.
Stephen, is there an animal model that you can utilize to test the direct relationship between infection and microcephaly without other potentially confounding factors?
Yeah, there is some mouse models in which they are using. There are actually a few papers showing these mouse models and the consequence of the infection. It's just a little tricky because the effect in the mice is different from the effect in humans, of course. And then one of these papers, for example, you don't have only the malformations in the brain. You have like a reduction in the whole animal. So, of course, it's important as an approach, as a different approach, but I think that I have to combine what is being done with the mice with what can be done with these human models.
Thank you. Are there any other questions or comments? One last. Otherwise, we thank you very much for your fine presentation.